Boy. No. Um, can people hear me? Yes, no. Yes, cool. And can you also see me on my screen? Yes, amazing. All right. I'm just gonna wait 2 more minutes and then or a few more minutes for anyone else who's coming. And then we'll start. Um, yeah, this is the first demo session, well, demo class of the new session that we're doing. Trying some new stuff out here on my iPad, so let's see if that works. Um, yeah, so if you guys want to grab yourselves like a glass of water or anything, go ahead. Um, and we'll begin shortly. I can also hear students. Yes, I can hear people. Um, but you can also write your questions in the chat. I think that might be easier, or you can raise your hand up or interrupt me at any point you want. This is supposed to be interactive. At least that's what I want it to be. I want this to be interactive. I want this to be something where there's a bit of back and forth. But well, let's see. That kind of just depends on the confidence you guys have. All right. Anyways, okay. Let's begin, shall we? It's 9 o'clock for me. Well, it's 9 o'clock, which for me it's 9 o'clock, which means it's 1 o'clock for you guys. So I'm gonna begin. First, before I start, I want to give a brief introduction of by myself. So my name is Daoud, and I did my O levels and A levels in Pakistan, where I took like 15 subjects in O levels, and I took five in A levels, and I don't know why I did that to myself, but I did it. And then I went to Imperial College London, and I did my undergrad and masters, so masters in engineering, in bioengineering at Imperial. Then I was unemployed for 7 months. When I say unemployed, I was just waiting for my visa to be approved. And then I started doing my PhD on a project between Imperial and Caltech. And I'm also a visiting researcher at King's College London. So the project I work on is on the nexus of like neuroscience, synthetic biology, bioengineering, and ultrasound imaging. So yeah, like what I do is pretty cool. At least I think it's pretty cool. That's why I'm doing it. Also very, very annoying. I'm making some drugs right now for treating stroke and epileptic patients, and that's going to be a spin out. So there's a lot of things happening right now. And now we also begin this A levels biology classes. Which, oh my god, this stops sharing my screen. This is a problem with Zoom. Every time I start talking and I forget about my screen or putting my finger on my screen, it stops sharing automatically. It's like it hates me or something. Um, okay. Somebody asked, how hard is it compared to O levels? How hard is AS level compared to O levels? I would say the jump isn't that bad. If you remember a bit of your chemistry, a bit of your biology, it should still be pretty straightforward. I think at least we'll try to make it straightforward. Because the thing is, like, people always assume that with biology, there's a lot of memorization or rote learning in other ways. And I do think there is a bit of memorization. Like, yeah, like you need to know the names of like certain enzymes, right? But there's always logic behind why things are happening. And what exactly is the rationale behind like a certain chemical reaction or not. And so that's like in today's demo class, like at least I'll try to give you a bit of insight as to why things are connected the way they are connected. And yeah, obviously, like you'll need to know, like, oh, the heart has four chambers. Like that, there's nothing I can really do about that. But there is a way to like explain that such that that okay, we have oxygenated blood and we have deoxygenated blood, and then that's why we have two halves in the heart, where one of them is pushing blood to get oxygenated, the other one is gathering all the deoxygenated blood, and we can work and understand how the pressure dynamics work in the heart when it's pumping in your systole or diastole, and why then we have four chambers. So it's easy to just say like, oh yeah, the heart has four chambers, but the rationale behind like why it has four chambers is, evolutionary. It also has to do with pressure and elasticity of the fibers, the blood pressure and the elasticity of the fibers in the muscle. And also, just interestingly enough, this is something you may or may not know about in your classes later on, but the heart cells, they're myopic, which means they don't need a signal from the brain to contract. When you grow like heart cells on a petri dish. So a petri, you might be aware of what a petri dish is. Um, it's just like a circular dish with a flat bottom where you grow cells, 2D culture. Um, so when you grow heart cells on a petri dish, you see that they kind of already have the contractibility, So that's why we call heart cells myopic. And there's a lot of cool things in biology like this. And I wouldn't say this is just biology. Biology is not linear. I would say biology is very, very interdisciplinary, because a lot of the big discoveries that happen. For example, you guys might have heard of a certain someone making AlphaFold, which is like this technology, this AI predictive technology that predicts the structures and folding of the protein. That is a very computational project. That in itself is a very computational project. It has nothing to do with like traditional biology of like, dissecting frogs or something like that. But that is very vital to biology, right? And that's what led them to winning the Nobel Prize in 2018. Also, I think they won it in chemistry of all things. So what I'm trying to say is sciences are always interdisciplinary, and you'll find out today, I guess, to some extent, what with biology and how it's interdisciplinary and so on. So let's start. Okay, so the first thing I'm gonna introduce is everything around you is made out of what? Elements, right? Everything around you is made out of elements. You see in your daily life, you see oxygen, you see carbon, you see hydrogen, and you don't really like interact with them apart from like oxygen, but you yourself are made out of carbon and hydrogen and nitrogen. So there's a lot of elements in the periodic table, but in living things are primarily made out of like four main elements. Like 99% of them is carbon, nitrogen, hydrogen, and oxygen, right? That's the few key elements that make up living things. And the interaction of these few molecules towards these few atoms, along with like additional ones, some additional ones, is what leads to a lot of complex chemical reactions, complex interactions, and so on and so forth. So if you think about yourself, if you think about you, human being, you are made out of what? Come on, somebody open your mic or like write it in the chat. Like, what are you made out of? Cells. And what are cells made out of? Mimi says cells. What are cells? Water. So you yourself, to a large extent, are made out of water, right? Even your own cells, individual cells, 80% of their content is just water. So cells, water. What else are you made out of? What else are you made out of? You said cells. So cells have water. What else does it have? And these are terms you might have heard of, like, DNA, protein. So DNA is what? DNA is a combination of nitrogen, carbon, and hydrogen, right? And oxygen, like organelles. And organelles are what? Organelles are made out of proteins. So Ismail says proteins. Organelles are made out of proteins. Genes, well, no. Genes is just the segment that codes for information. It's kind of like a word we throw around, but like the actual thing is DNA. But yeah, genes too. Sure. So you're made out of protein. You're made out of, to a large extent, like not. It's not just your muscles, by the way. When you think protein, you don't think about your muscles. You should think about what someone else said. Mimi said, like organelles. You should think about like protein channels in the membrane. And if you've taken biology before, all of this will make sense to you. But if you haven't, then don't worry. We'll get there eventually. So you're made out of protein. What else are you made out of? You're made out of carbohydrates, right? Everyone says carbohydrates. Oh, of carbohydrates. Let's do this. We'll get there. I'll guide you about syllabus and paper, but I want to start the topic of, this before we get into like what the syllabus is going to look like and what the papers are. And then you have fats, right? Fats. And to a large extent, people assume that carbohydrates are like a big chunk of your body composition, but it's actually like around 1 to 2%. It's not even that much. And the thing is, carbohydrates are what you need for instant energy. Most of your energy is stored in proteins and fats. So when you look at like these key things, when you look at them, you realize that all of them are just an interaction of carbon, nitrogen, hydrogen, oxygen, and some other elements, right? So what this tells us is that the interaction of these atoms and these bonds is what leads to life, right? And what interactions are we talking about? What interactions are we talking about? You know this. You've seen your Instagram gurus talk about this. Fitness gurus. You've seen any older person who has diabetes, or like you tell them to go to the gym and they're like, oh, my metabolism isn't fast enough. What? So it's the same thing. Metabolism is what I'm trying to get to, which is people throw this word around, but metabolism basically is all the chemical reactions happening in cells and living organisms. Because I'm trying to connect this for you, which is elements, carbon, nitrogen, hydrogen, oxygen. All of these is what makes up living organisms. So and their interaction is what leads us to do activities, to breathe, to reproduce, to create waste like excrement and so on and so forth. So what is the term that encompasses all these interactions, all these chemical reactions happening in your cells and living organisms? It's metabolism, right? People might say this as a normal thing, but like think about it. Your hair is constantly growing. That is metabolism. That is a reaction happening in your body, right? Or you are breathing. That is also metabolism. That this is all part of the same umbrella, right? Which is like everything comes under metabolism. There's a base metabolism, which is you do nothing, you sit on a chair, and that's like you're respiring, your hair is growing, your nails are growing, you're doing X, Y, Z, and that's like your base metabolism. But what I'm introducing metabolism is more so as a concept, that the interaction of these molecules, these atoms, these elements, right, is what leads to life. It's what leads to living things doing what they do. And so there's two key sub reactions in metabolism, right? If you're please mute yourself. Yeah, please mute yourself. So there's anabolism and there's catabolism. Guys, for context, I'm gonna say this now. I will talk about the syllabus towards the end. So let's put a lid on that for now, because I feel like we're in a flow. At least I'm in a flow. I want to keep on talking about this. So anabolism and catabolism, right? What is anabolism? Now, keep in mind, these are things you need to know for your AS level. What metabolism is, what anabolism is, what catabolism is. And you'll see as we go on in the syllabus or in the course why they're so fundamental to understand. Because what I'm trying to do is create this map in your minds, which is anabolism is what? It's when small molecules. So this is just a molecule I'm drawing. It's nothing complex. When small molecules, they come, I'm just gonna mute. Oh, how do I mute? Guys, if you're not muted, please mute yourselves. Okay. I have failed. Oh, okay. But you can hear me now. Cool. Can you hear me now? Yes? Ah, perfect, perfect. Okay. So anabolism, right? So we're talking about anabolism, and I said anabolism is when small molecules, maybe we should change the color slightly so it's like more clear. When small molecules, they come together and they form bonds and create a larger molecule. Right? So you go from something small, smaller molecules, and you create a large, complex molecule structure, right? That is anabolism. Yes, so when you're making exactly, so look, this is already clicked for Ryan, right? So you're going from tiny amino acids and you're creating a big protein. Again, don't be afraid of these terms that I'm throwing around, like amino acids. If you've taken biology before, it will kind of make sense. If not, we're gonna cover this. But the key concept that you need to understand is making anything in your body is pretty much anabolism. And the other key thing to kind of note right now already is when you do this, the creation of these chemical bonds, right? It's called a specific reaction. So anabolism is when you synthesize something, right? But the reaction, the name for the chemical reaction that leads to the creation of these large complex molecules is actually called a condensation reaction. And do not worry, I'll go into more detail on condensation reaction in a later video. For now, this is what you need to know, right? And I'll explain why it's called condensation. Sir samajh nahi aaya. So small molecules, they come together, correct? And that you understand? Yes, small molecules come together and create a large molecule. Do you understand that? That's pretty straightforward. Yes?
सर
Yeah.
वो कंडेंसेशन वाली बात समझ नहीं है.
Acha. So you understand that, right? For condensation, what I'm saying is when you create these chemical bonds, right? There's a name for this reaction. Going from A to B, going from state A to state B. The name for the reaction that leads to this, so anabolism is the whole underarching thing, but the reaction that leads to this, the chemical reaction that leads to this, is called a condensation reaction, right? And I'll explain why it's condensation slightly down below. But for now, what you need to understand is you condensed a lot of small molecules into one big molecule. Like think about it like this: you have this whole structure, and then you're just kind of condensing it and making it into like a more compact big thing. So that's you can think about like condensation reaction in that way. But don't worry, it will make sense once I start drawing out like the chemical structures. We'll get there. Catabolism on the other side, so that's anabolism. Catabolism on the other side is, yes, condensation reaction is the one that releases water as a byproduct. Yes, but I want to get to that when I draw the chemical structures. Catabolism, on the other hand, it's kind of very easy for us in Pakistan or like anyone who speaks Hindi, Urdu to understand. But catabolism is going from a big structure to a smaller structures, right? So Kartna, right? Catabolism, Kartna, right? You cut, so you have this big structure. Let's say this is your structure, and then you break down these bonds between the small units, and you go back to your initial separated smaller molecules. This, the chemical reaction that leads to this, the formation of small molecules from a large one, is a hydrolysis reaction. Now, for now, I'm only introducing these as terms, but I will explain them in a second. Do not be phased. Okay. So, in reality, you can think of this as exactly. So, Ryan says glycogen to glucose is a catabolic reaction. Yes, exactly. Deamination of amino acids, well, yes, is a catabolic reaction because you're going from something big to something small. And in a lot of ways, fats to fatty acids and glycerol is again a catabolism reaction. So you're getting it. You're getting it. You understand that anabolism and catabolism are inverse of each other, right? Right? In one, you go from small subunits to a large complex molecule. In another one, you go from a large complex molecule to small subunits. And so, exactly, someone said hydrogen peroxide into water and oxygen. It's an example of, well, it comes under the overarching, metabolism arc. And yes, it could be catabolism depending on how it's happening. But yes, you're right. Like all of these things going from big to small is like, catabolism. Okay. But this is something that you can probably look at in your books or in your classrooms. The thing that I want to like point out right now is, have you ever thought about what is happening to energy? Because remember, I said to you guys that metabolism and all the chemical reactions happening in your cells and living organisms, it is basically, it's not, it's not created or destroyed. It's not, but it is transferred, right? So anabolism and catabolisms are inverse of each other, but they're also constantly communicating with each other. How are they communicating? Somebody said ATP. Yes, exactly. They're communicating through ATP. Now, the thing to understand is that anabolism, so you're making a large structure. Usually, when you're making a large structure, this is a way of explaining. Sorry, I'm just gonna draw a line. Yeah. So let's say this is a graph that I have. And on this side, I have energy, right? And on this side, I have time or reaction, right? My substrates, so my reactants, they have this much energy, right? They start off here. The reaction takes place. The reaction takes place, and so I'll label this as reactants, and then my products have this much energy. So I basically went from this to this. And somebody said energy is not created or destroyed. So what is happening to this energy? Because now I've created something from something that had higher energy, and now I have less energy. What is happening to this energy? It's going where it's being released, right? That could be in the form of heat, that could be in the form of chemical bonds, that could be dissipated to the surrounding, lost to the surrounding. It could be different things. This we call free energy. Some of it's free and some of it's not, but the usable one is free. And this type of a energy plot, right? By energy plot, I mean this type of an energy dynamic is called what? Exogenic reaction, right? Exogornic reaction. Oh, yeah, pronounced this wrong. So exogornic reactions. Yeah, there you go. Well, yes, so here's the thing. Cool thing, right? We are talking about energy. Exothermic reactions, while close in concept to exogornic reactions, are not the same thing. Exothermic means to do with heat. Exogornic isn't just heat, it's everything. It's any energy. So yeah, most exothermic reactions are exogornic as well, but it's not always like a one-to-one thing, right? The second case we need to look at is this. And this is when you start off your products, your reactants at a lower energy. So this is your reactants. And then your product has a high energy, right? So now you're starting off with something that has low energy and ending up with something that has a lot more energy. There's an energy increase. So what are you doing? You're actually supplying free energy. You're supplying free energy, right? You're going up a hill. You're going adding. You're going up a hill. It's incredibly difficult to do that. What is this reaction called? Well, what is this term? This energy reaction? It's called endogornic. So one is exogornic and the other one's endogornic. And again, what I want to understand, yeah, exactly. Absorption of energy is happening. Absorption of free energy is happening. Because look, endothermic reaction has to do with heat. Exothermic reaction has to do with heat. But endogornic reaction has to do with the transfer of free energy when you're inputting free energy into the system. And exogornic reactions have to do with the release of free energy from a system. And all these things, the release of free energy, the absorption of free energy, this is all communicated through ATP. And this will make sense in a second. This will make sense to you in a second. I'll tell you why it will make sense to you in a second. But first, we need to understand something else. We need to understand something very, very, very important. Water. Why? Because look here, what did I say? What did you guys say actually? Cells have water. 80% of the content of cells is water. We are like what, 65% water, right? Water is very important. Water's properties are part of your syllabus, actually. Understanding why water is the way water is, right? And for that, what are we going to do? We're going to look at atoms and elements of water. So anyone who has a brain cell active right now, please tell me what is this formula of water? H2O. Good job. H2O. Good, good. What does this mean? This means one oxygen is bonded to a hydrogen on each side. Now, this is something you learn in your chemistry. Maybe you've learned it in O-level chemistry. I don't remember my O-levels. It was like such a long time ago. But maybe you did, maybe you didn't. But you will learn it if you're taking chemistry. And I'll try to explain it very, very briefly to the extent that you need it. Everything has electrons, right? Correct? So oxygen has how many electrons in its outer shell? It's in the sixth table. Exactly, six. So 6 electrons in the outer shell. If you know rudimentary bond sharing, then you know that hydrogen has 1 electron and it's sharing an electron with oxygen. Same on this side. So that means this is, a complete molecule. But you need to realize that while you formed a covalent bond with oxygen over here, hydrogen, hydrogen, oxygen, these 2 electrons are still present, and we draw them as lone pairs. Lone meaning they're alone, they have not formed a bond with anyone, but they're still there. Lone pair. Pair is 2, right? Lone is alone. They haven't formed a bond with an atom. They're still on the oxygen. They didn't disappear. The other important concept to understand here is, now this is where it's like very beautiful, and this is where like a lot of pretty much all chemical reactions kind of depend on these properties. Oxygen is a greedy motherfucker. What is it greedy for? It's greedy for electrons. Which electrons? The electrons it's sharing. Oxygen has a stronger pull. So this bond, every time you've seen a bond between any 2 molecules, you've already assumed that it's shared equally. Well, this isn't communism. This is capitalism. Nothing is shared equally. Nothing is equal. Oxygen is a much bigger, much stronger, much more greedy element. What it will do is it will pull this pair of electrons towards itself. Ever so slightly, not too much, but enough. And we say the tendency for an atom or an element to attract, to want electrons is called electronegativity. And you learn about this more in chemistry. But I'm trying to make this clear to you that biology is very interdisciplinary. There's chemical reactions, there's physics. This energy diagram is basically physics or thermodynamics. Now we're talking about chemistry, and all of this is related to biology because biology is life, and it's because of the interactions of all these things that led to life. So I will now draw this lone pair more towards the oxygen on both sides, right? Because it's pulling it more close to each other. So it's not an even share. It's an uneven share. So now that I'm taking the electrons more towards the oxygen, what is happening to the oxygen? Electrons are negative. So the oxygen becomes partially negative. This is the symbol for partial. It becomes partially negative. Hydrogen, it becomes partially positive. Why? Because the electrons have been moved away a bit. So your oxygen is partially negative and your hydrogen is partially positive. Now, this is where it gets interesting. Water molecules are all like this, and we say that they are polar. Polar meaning they have charges. They have this partial charge. By the way, what I'm explaining right now is very, very rudimentary in A levels. Like, this is something when you look back on, you'll be like, oh wow, this is the most simplest thing of all time, because it'll just make sense automatically, right? Water is polar. Polar, it means it has charges. Why does it have charges? Because oxygen doesn't like to share its electrons with hydrogen equally. It will always pull it towards itself because it's very greedy, because it's more electronegative, right? And so that leads to the creation of these charges. Now remember, in a glass of water, there's not just one water molecule. There are well, a lot. There's a lot of water molecules, right? There's a lot more. In one mole, there's like the Avogadro's constant number of molecules. Like there's a lot of molecules, and all these molecules have these charges on them. But now you can use your brains. and tell me if there is another water molecule, so partial negative, partial positive. And let's say there's another water molecule, partial negative, partial positive, partial positive, right? And let's create another one. What is going to happen? Do you have negative and positive charges? The negative charges are going to be attracted to the positive charges, right? The positive charges are going to be attracted to the negative charges. These are your intermolecular bonds. This is an example of an intramolecular bond because it's a covalent bond, right? And so this is where I introduce you to something very fundamental to biology, which is hydrogen bonding. Hydrogen bonding is when hydrogen forms a bond with fluorine, oxygen, or nitrogen. Fun or fun, right? This is what hydrogen forms bonds with. And whenever hydrogen forms bonds with any of these molecules, it leads to an uneven charge distribution. Uneven positive and negative electrons, right? So you have hydrogen, fluorine, partial positive, partial negative. You have hydrogen, nitrogen, partial positive, partial negative. You have hydrogen, oxygen, partial positive, partial negative. Why? Because these elements are very greedy. And so they pull the electrons towards themselves, leaving hydrogen empty. If there's lack of negative charges, it means it must be positive now. Unless it's neutral. Yeah, like if you had a negative charge, I took it away, now you're positive compared to what you were earlier. And these partial charges leads to the creation of these intermolecular forces. It's very simple, actually. It's very simple. Because this is hydrogen bonding. This is your hydrogen bonding. And this is a phenomenon of hydrogen bonding. And it's amazing. Because these two things, this intermolecular covalent bond and this intramolecular covalent bond and this intermolecular attraction of partial positive and partial negative, like polar charges, is what gives water its amazing properties of what? Of being a solvent. Why do things dissolve? Now, this is an important property. Please take note of this, right? Because in your A levels, they will ask you about the properties of water in different contexts, and you would have to connect them to yourself, connect them somehow, right? And I'm trying to create that image for you, at least, yeah. So why is it a good solvent? Okay, let's look at salt. Salt is NaCl. Salt will have the same thing, right? Salt, when you dissolve it in water, Na is positive, chlorine is negative. You've learned this in your chemistry from O levels, right? It's, positive ions, negative ions, correct? Now, water, this is water. I already explained to you why there are negative and positive charges in water. So partially negative, partially positive. This allows the interaction of this, and the negative of chlorine is attracted to the positive charge in the hydrogen. And so this is what allows it to be soluble. This property of interaction of these ionic charges with the partial charges of the water molecules is what allows the solubility. Now think about it like this. Oil, lipids, oil, fats, right? Lipids. They're not soluble in water. Why are they not soluble in water? Because there's no charges from the lipids to interact with the charges of water. Right? Take anything else. Take magnesium chloride, right? The magnesium positive two charges will interact with the negative charge of the oxygen. And the chloride would again interact with the positive, the hydrogen. This is what allows solubility. If you ever in your life have to think about what is soluble and what is not soluble, think about it like this. Is the molecule going to exist in ionic state? Is it polar? If it's polar, yes, it will dissolve, right? Do all hydrogen bonding molecules act as solvent? Yes. Yes, they do. Because you can infer it from this concept. If you are dissolving something polar, by polar I mean something with charges, right, into something with charges, positive and negative, then they are soluble. If you are dissolving something that's non-polar, so non-polar, this is polar, this is non-polar, polar, yeah. If you're dissolving something non-polar into non-polar, then that will work because they're both don't have anything, the soluble. But what doesn't work is this cross thing. This doesn't work. This is not possible. You can't dissolve a non-polar compound in a polar solvent, and you can't dissolve a polar compound in a non-polar solvent. They just don't interact like that. Exactly, and this is why oil and water don't mix. Now, my question to you is, and this is, maybe you can take this as homework and tell me about this, because I want you to think. I want you to use your brains. Don't just ask for answers. Um, think about it. If everything has to be polar in order to be dissolved in water, our blood, the major component of our blood is water. That means proteins are polar, XYZ is polar, whatever. But there's a lot of non-polar compounds. For example, lipids, fats, like I said, we said in the beginning, fats make up a big chunk of us, like 16% almost. So how are lipids then being transported in the blood if they can't dissolve in the blood? Because blood is polar and lipids are non-polar. This is something you should think about, and let me know later. So I hope that the idea of why things are soluble in water now makes sense because of the interaction of these charges, right? But it's also the same concept of these intermolecular forces and intermolecular forces that leads to other properties of water. Number two, let's talk about number two. Water has specific. I mean, okay. Also, when I said solvent, keep in mind that also includes medium transport of medium transport, right? So medium for transport, because like I said, blood. So like this is just a inferness of the fact that I said solvent. So it allows for transport of things. The other one I want to look at is uh, let's do specific heat capacity. So you've definitely learned this in O-level chemistry, and you will relearn it again in A-level chemistry if you're taking it. But very briefly, specific heat capacity is the energy required to raise 1 degree of something, of 1 kilogram of something, I believe, or is it 1 mole, or something like that? Yeah. So to raise the temperature of something by 1 degree, the something is like to raise the temperature of 1 kg of something or 1 mole of something by 1 degree. That's what specific heat capacity is. Now, look at the structure of water again. And this is where it gets interesting, and this is where it gets interconnected. You understand why there are partial negative, partial positive charges, right? So, and then that's the main two. Let's just draw it like that. Okay, so if I'm going to increase the temperature of water by 1 degree, I need to provide energy to all these bonds. And there's millions of them. There's going to be another oxygen over here. These water molecules need to overcome the forces that are keeping them together. And we need to supply them with energy so that they're more kinetic. Kinetic meaning they're moving. Now, this is physics, right? And so, a very good diagram to explain what I mean by this is this. Every system has internal energy. Internal energy is potential energy plus kinetic energy. And I'm assuming you're all people who did physics in O levels, so you should know what potential energy and kinetic energy is, right? Kinetic energy is the energy you have through your movement. Potential energy is the energy you have at rest. The connection of all these things is what gives you internal energy. Think about it like this. Even if think about it like a box, a solid box. There's things moving around it. Like the atoms are vibrating. They're not moving, but they're vibrating, right? That vibration is kinetic energy. And the fact that you've stored the box on like a shelf and the box can fall and then transfer energy, that has potential energy, right? When it's stored on a shelf. So there's kinetic and potential energy present at all given times and things. So when I have, like, let's say temperature on this side, like on this graph, and let's say this is the time or energy supplied. If I am going from negative, this is negative. So when water is frozen at negative temperatures, right? So solid, let's say this is 0. So I'm going to draw this in a different color. So I'm going solid. Okay, so let's call this 100. Okay, and this is 0 degrees. Okay, so water is solid when it's negative. It starts to freeze or melt at 0, depending on which way you're going, right? And then it starts to evaporate, boil, turn into steam, gas at 100. Correct? The specific heat capacity of increasing the temperature of water by 1 degree, where is that happening? Where's that happening? Come on, guys. Is it happening in A, B, C, or D? Where is the temperature increasing in this? B and D. Good job, Ramin. B and D is where the temperature is not increasing. It's not increasing in B and D. It's increasing in A and C. At B and D, come on, guys, look at this. The temperature is constant. It's not moving anywhere, right? It's not going anywhere. It's constant. The temperature is constant. Constant. Time is going on. Time is going on. But please change at phase. Exactly. Phase is changing at B and D. That's why I said freeze and melt and evaporation. But the temperature isn't changing. So where is your specific heat capacity? On A and C. This is where you can deal with specific heat capacity. So specific heat capacity is basically the change in energy. Energy change it. So, and the equation for this, which you don't need to know, but I'm just giving it to you, is change in energy equals mass times the specific heat capacity. Every substance has a specific heat capacity times change in temperature. This solves for the amount of energy you needed to raise the temperature of something by 1 degree, which is what I said, right? 1 kg of something by 1 degree. Correct? Yeah. Why is this important? Because the energy required to change something by 1 degree, especially water, is quite high. It's quite high. And it's high because of these intermolecular forces, right? Intermolecular and intramolecular forces. Because of this hydrogen bonding. It's high because of this. Why is that important? Think about it. Contextualize this. It's difficult to change the temperature of water by 1 degree. Why is that important? Strong IMFA between molecules. What is IMFA? I may be too old for this. Oh, no. Humans are made out of water. Bodies are made out of water. Everything is made out of water. If you think about yourself, if my water body, like you said it in the beginning, I didn't say this. I'm just trying you to connect it, right? I'm just trying you to connect this. If I increase, if the water in my body changes temperatures too quickly, fluctuation, then my enzymes might die. I might die. I don't have a stable environment. Think about a lake. If the temperature of the lake is constantly changing, the fish never have a stable environment. Do you think things can live in an unstable environment? You live in Pakistan. Like, think about it like this. Can you live in an unstable environment? You can't. It's shit. So if water changes temperature like like in a fluctuating manner and very easily, or every time like the outside temperature increases by 2 degrees, I start like boiling inside because my water like it's not it's like the heat capacity if it's quite low specifically, so very little heat is required to change its temperature. Then water isn't a good medium to transport anything, and water isn't a good medium to hold any living organism. This is why specific heat capacity is important, because it allows for a constant, stable environment. Okay, next one. Uh, Latent heat. Okay, so there's another concept, which is in these phase changes, right? This is your specific latent heat of evaporation, right? Why is that important? That is important because look at this. This is the diagram I drew. When I apply roughly around 40.7 kilojoules per mole of energy, I start to break these bonds. I start to break these bonds. When I break these bonds, what happens to this? It becomes a gas because there's nothing holding it to the next water molecule. There's nothing holding it to the next water molecule. It's no longer liquid anymore. I've supplied a small amount of energy, broken this into, broken the bonds, and then gone into like a different state. Why is that important? Why is that important? That is important for what? For cooling. Living organisms, they have water droplets all over them. I am sweating right now because my room is really, really hot. And there's no fan or AC. So, you know, I have water on my skin. To break these intermolecular forces, you don't need that much energy. To break these intramolecular forces, you need a lot of energy. And that's why water is like, such a good, has such a high specific heat capacity. In fact, I think it has the highest specific heat capacity of any known substance, but double check me on that. But for cooling, for in this phase change in D, it doesn't require that much because you're not changing the structure of, you're not breaking HOH, you're not breaking these bonds, you're just breaking the interaction that they were having because of the positive and negative charges of adjacent water molecules. You're just breaking these ones. And so when you are hot, when the organism's hot and there's water on top of it, you boil, well, you, yeah, you boil the water basically, it kind of evaporates, right? It goes away. When it evaporates, it does what? It takes energy from your skin. This is the water droplet, right? I'm supplying it heat from my skin and it becomes gas. So the energy is being transferred from my skin, which is hot, to the water droplets on my skin and then evaporating. I'm cooling down. Life is always trying to preserve itself. Everything is connected. The properties of water allow us to have a stable environment inside so we don't fluctuate all the time with the slight change in the temperature outside and kill our proteins and our cells. Water is polar and allows for things to be dissolved in it. Water has a lower specific latent heat of evaporation because it allows us to cool ourselves down. It's all possible because of the interaction of these molecules. There's two more properties of water: cohesion and adhesion, and they have to do with the interaction of water molecules because of these bondings. The same bondings I've taught you. There's nothing else to it, and I will explain that in the next class. And then we will come back in a full circle to why this is important for energy transfer in ATP. Yeah. Okay, so I will stop here for today. Um, continue in the next session. More properties of water. And I hope after that you can have a full appreciation of why biology is so, interconnected with everything. Okay, so what is your A level AS syllabus, or what is your examination method? So there's 3 papers in A levels. There's always going to be 3 papers in A levels. Paper number one, AS levels, I mean. Paper number one is your MCQ. You have 40 MCQs, just like you did in your GCSEs, but you have slightly longer. You don't have 16 minutes. You have 1 hour and 15 minutes, I believe. You have paper two, which is your theory paper, just like you did in your O levels. You have an MCQ paper, you have a theory paper. This is again worth 60 marks. You can have up to 4 to 6 questions depending on how they mark the questions. 40 marks. 4 to 6 questions. 60 marks. And then finally, you have your P3. P3 is a practical paper, no more ATP. It's a practical paper. Your schools will arrange that. If you're a private candidate, they will arrange that for you. But basically, you will learn in your classrooms a few important techniques. For example, like how do I dissect an onion and put it on a slide and examine it under the microscope? How do I draw what I see under the microscope? So that could be like high resolution, high power resolution drawing, or it could be low power resolution drawing. And those are things that we can learn outside of like a physical laboratory in the syllabus. So how to do a serial dilution or like a simple dilution, and it's all very simple. But then you have to do the practical. Every year, this stuff gets leaked. I don't like it. I don't think it should. But the practical is going to be simple. You will go, you will examine a plant cell or an animal cell under a slide and just draw it out, or they'll ask you to like mix some enzyme with another enzyme and look at the oxygen coming out. But that will make so much sense to you because you're understanding, at least the way I'm trying to build it would be pristine, right? So there's three papers: paper one, paper two, paper three. Simple stuff, not really that complicated. It's literally the same pattern you've learned in O levels, right? You'll have 40 MCQs and you'll have 4 to 6 questions to write. The next is syllabus. So I want you to focus more on the learning aspect, of A levels, because it's very important as a foundation if you want to have higher education. The syllabus in AS levels is 11 chapters roughly. And the first few chapters, of which chapter 2 is what I started off with, which is biological molecules, are your cells, your biological molecules, your enzymes, your cell membrane, and your DNA synthesis, central dogma. There's a few 2 chapters on that. Then there is transport. So I can write this all down actually. Why not? So you have cells and organelles. And in cells and organelles, it's literally the same thing you've learned in O levels, but with a bit more functionality attached to it. Number 2 is biological molecules, which is the key things that make up living things, right? That's your amino acids, proteins, carbohydrates, fats, lipids, and water. And with which we started water. Because water's properties connect everything. Number 3 is your enzymes. What are enzymes? What is your Michaelis-Menten equation? What is an enzyme affinity? It sounds complicated when I say some foreigner's name like Michaelis and Menten, but like it's simple. It's simple. Don't worry. Then you have cell membrane, right? And this is where you learn a bit more about the way the cells interact with one another and so on and so forth. Then you have 5 and 6, which are on mitosis, the cell cycle. So mitosis and the cell cycle, mitosis and cell cycle. And then you have 6, which is on DNA and RNA translation, transcription. So I'll just call this central dogma, right? Then you have 7 and 8, which is transport in plants and transport in animals. Then you have gas exchange. So how do your lungs work? Gas exchange, and the heart. So that heart is in the transport in animals. Then you have what was number 10? I think it's infectious diseases. And then lastly, you have immunity. Now, one of the things I want to try out this year, so these are the chapters you have. We've already started with 2. You might be asking me, why are we starting with 2? Because I think 2 is just makes it simpler. Because if you know what a protein is, and you know what water is, and you know how these interact, you will learn cell and organelles much better, and you'll learn enzymes much better. And this is an important foundation to have. Something I want to try out this year is I do work in bioengineering, and I have a lot of friends working on very cool projects. And whenever there's something where I can think of like one of my friends from Imperial or Caltech or Oxford or Cambridge that can present to you guys on an interesting topic, just to like engage you rather than to like just make you learn A levels, that could be something I want to like try out this year. So, for example, if we do immunity, my friend literally makes antibodies to treat cancer, and so it will be a good talk from her. Or if we work in like transport and plants, I have a friend in Okinawa now who works on using plants to synthesize like medicinal products or like biological products or any kind of product you can think of. Like you can create plastic by using plant as your foundation, as your organism of choice or a biofactory, right? So A levels is pretty simple. There's 3 papers. There's 11 chapters. we'll try to finish the syllabus by, I don't know, December, Jan, Feb. Last time I finished quite early, but then it really depends on how fast we're going. But it will be finished in time for a past paper session. Maybe occasionally here and there we can add past papers in the mix. So when we're doing like chapter 2, we'll finish it off with a few questions to kind of like put it, because like if you don't practice, you won't learn. Based on my schedule, it'll be 2 to 3 classes a week, but at the moment it's going to be 3 classes a week. Classes are supposed to be 3 classes on Mondays, Thursdays, and Saturdays. The reason it's kind of spaced out and not together is I believe that if I give you information over 3 days, you won't internalize it. Your neurons and synapses won't have enough time for adjusting to the new information and just getting like overwhelmed. So Monday I teach you take a break, Thursday I teach you take a break, and Saturday and then you think about it. So recalling actually is important. So Monday it should be 9 p.m. to 10 p.m. Thursdays it's 9 p.m. to 10 p.m. and Saturday, well, I don't remember what I said, but it's like 12:30 p.m. to 2 p.m. Or it's either this. Double check. I'll double check this, but I think it's 12:30 to 2 p.m. Pakistan time. Yeah. So this is the schedule. I'll send more information about this in the group chat. Actually, they all will. Do note that I am in London. My time zone, there is a time zone difference. So like for me, this is like quite early in the morning, but it's fine. It's 4 hours right now because we're on British Standard Time. But when we move to GMT, it will be 5 hours. So when that happens, and that will probably happen in October, the class timings might have to be adjusted because then it would be 10 to 11 for you guys or something. So we'll see how that goes. But that's a while from now, and there's nothing I can really do about that because it's just time zones that they it's funny that how they make those time zones. Yeah, so this is 9 p.m. Pakistan time, 9 p.m. Pakistan time, 12:30 p.m. Pakistan time. This is all Pakistan time. This is all for Pakistan. So this is the information on how we're going to do this. I'm going to expose you to top research that's happening in the world. Imperial is number 2, so pretty good. And there's a lot of cool things happening. And not only that, I hope that you guys can connect the dots between biology, chemistry, physics, and how all of it is related. But there will be periods where it's kind of like boring-ish. I hope I don't make it boring, but we'll see. We'll see how it goes. And I hope you enjoyed today's class. Any other questions on what we covered or the syllabus or the papers?
Sir, how many marks is practical?
Practical is, I believe, 40 or 60 marks. I will double check that. But yeah, it's the same. Yeah, 40 marks, yeah, as MCQs. In your MCQ paper, I will say that if you can consistently get 35 or above, you have a good chance of getting an A. And the reason I say this is because people practice a lot. The MCQ paper can be very difficult, and then you may get like 27, 28, 29, 30. But 35 and above is like you are kind of like putting yourself in a good spot because that is what the threshold for that paper usually are. Sometimes it's like 39 for an A, So I remember doing past papers in my own time when it was like 39, 38 when I was doing past papers. For theory papers, 45 to 50 plus is what you should aim for to get an A. Practical, same thing, 30 plus is what you should be aiming for. this combination would kind of allow you to say more confidently that you might get an A, and that is something you will probably learn as you do more and more papers and practice questions, and you'll see what you're making, what you're getting wrong, and like how often are you getting it wrong, and so on and so forth. A levels isn't complicated. At least AS level isn't complicated. I do believe that wholeheartedly, especially with biology. I think it's like one of the most like it's one of the easiest A level subjects you can take. Don't Jesus says do like I say, not like I do, right? So when I was doing my A levels, like I just worked in the last 2 months and I got an A star and shit. Like, but don't do like what I did. Study. Do it before. It's really intense after. And for biology in particular, I felt like I didn't really, do a lot of hard work. Like it's just like just came natural because it was quite simple. Like this is what a protein is, and this is how a protein works. Like, and if you know that, you can understand enzymes, and you can understand cell membrane, and you can understand ribosomes and how they're like, translating DNA, and you can understand how glucose is being transported and stored. Like it's very simple. Any other questions, guys? The floor is yours. But if there's no questions, then I will bid you guys adieu, and I'll see you guys in the next class if you are coming. Take care.